So what is nuclease protection footprinting? DNase I footprinting. So DNA I footprinting assay is used to study the DNA protein interaction, okay, and to identify DNA sequence which protein binds. What is the exact sequence of that particular DNA fragment where DNA is bind with the protein or protein is binding with the DNA. This is the same thing. It measures the ability of protein to protect radiolabeled DNA fragment from digestion by DNase I. It also measures what is the ability of any protein and how it is protecting it from the cleavage. And for this measurement we use DNA label with radioactive isotope elements. Why? Because that will give us visual identification. Okay, identification. So visually हम उसको आइडेंटिफाई कर सके. That's why we are using radio label DNA and fragment from digestion by the DNase I. And that particular fragment which can protect the DNA from the DNase I by the protein protection. So DNase I is endonuclease. It will cleave middle of somewhere in the DNA fragment with little sequence specificity. It have little sequence specificity. What does it means? It can cleave if there is little similarity seen by this DNase. So if we see in this strand, if there is their specificity is not so much, it's just two sequence, just see A and C. So their probability of A and C repeat will be so many in the single strand. So that's why it can cut very rapidly and anywhere in the DNA molecule. So for this DNA first treatment is carried out in the presence of manganese salt. Manganese and magnesium both are different elements. Here it is Mn, manganese. Manganese salt which induces random double strand cut. So it is helping or increasing the efficiency of the DNase I and produce blunt end because it is cutting double strand. You can see this is a double strand of the DNA. If it is cutting in endonuclease way means in middle of somewhere in the strand. So suppose it is cutting from here. So it will cut down from this way. So what is the fragment will form? Fragment will form this the exact blunt end. There is no single nucleotide will remain this way. So the DNA can form again in long. So there will be blunt end. So there is possibility that formation of again DNA or attachment of frequent attachment of DNA strand to each other will reduce by the blunt end fragment. So now nuclease treatment is performed under limited condition. So that's why, because it can cut anywhere in the DNA sequence, there is less specificity. So that's why we are using limited condition or low temperature and very little amount of enzyme. So we cannot cut so much fragment or cannot make so much fragment, which is not our ideal way to study this. We want only single cut at a fragment. At single fragment will cut single time. That's why we use a very little amount of this enzyme. So because it can cut anywhere, so its efficiency is high to in cutting, so we take a small amount to cut our only one position in a one fragment. Under this condition, each fragment receive a single hit. Each fragment will cut only at a one place, and this will generate all possible size, forming continuous ladder on the denaturing gel. How? This is very important line. If it is cutting at a only single position of a stand, it doesn't mean that all stand will cut from a specific site. No, it will cut anywhere, but at a one position only. If this DNA segment is cutting a only single cut, so there will be here, will be here, will be here, will be here, but only one will be cut is done, other are no. So if these there is a so many 100,000 of fragment are there, so one is cutting, suppose here, it doesn't mean that this is also cutting from here. No, all will cut differently. Anywhere the little enzyme amount is there, so enzyme found any place or any place to attach on that segment will cut down by the enzyme. So if this is enzyme is here, then cut from here. If in some segment this is here, so it will cut from here. If in some segment this is it is present here, so it will cut from here. So there is so many fragments have different places on that stand to have a single cut. So if we suppose, and then we do denaturing gel denaturation, so we get single strand of each fragment, and that will arrange according to the size, because what gel do, gel will separate the strand of DNA according to their sizes. So as we can say, suppose someone is cutted from this is not the specific, or this is not the sequence of cutting. This is sequence after the electrophoresis of that cuts. So it is look like a ladder. Ladder means a CD. These are the ladders to go up a stair. You have ladder to put a step on that ladder to go on the upstair. Hope you understand. So what does it means? So they form a ladder. Understand how the ladder is forming after gel electrophoresis. Because of the size wise separation, size wise differentiation of the DNA fragment. If their large size of fragment will move slow in the electrophoretic gel. If their DNA segment have small size, that will move faster. So small size will be here, and after that here. This will be the sequence of the DNA strands in the electrophoretic gel. So if a protein X binds to a specific DNA site, okay, that region is protected from the DNase I digestion. As we see, what we are studying here, DNA protein interaction. So on the DNA at a specific site, some kind of protein are attached and protecting them from the DNase I digestion. So that particular sequence or that particular part of DNA cannot get cut with the DNase I enzyme. So as a result, what happen? A gap or footprint appear. That gap is what? Gap is due to protein presence. And why? Because DNA 1 cannot be able to cut that part. So that gap or that foot is the what? That is protein present on that particular DNA sequence. So in the DNA ladder, the footprint is appear. We'll see footprint corresponding to the DNA region where protein X is bound. So footprint means there is a protein present. So footprint means something is present there. Someone is came here. That's why there is a foot who shows that someone comes there. So their foot is protein. So if you can see, if it this is the place where protein is present, so DNase I cannot cut this part of the DNA. It can cut from this, from near of this, but cannot from this part of the DNA. So if it cannot cut from this part, so this is in where protein is present. We are comparing one strand where protein is present and second we running two parallel gel in which one have protein in their DNA and one we remove protein from the DNA and then we run the electrophoresis. So that will be this type of result if there is no protein is present. And if there is protein is present, will show this type of ladder, and that portion, this specific portion is showing the gap due to presence of protein which cannot be get cut down, so that will remain here only, which will be higher in length because that strand is not cut down, because we only cutting from one place of the DNA sequence. So that will be the on that position that is not making or having participate in the ladder formation, and that we can see the gap where the DNAs are not able to cut down. This is the story of protection. Now it is not complete yet, so listen. In the assay, DNA fragment contain binding site is mixed with purified DNA X and DNase I. What we do now? We have to do some experiment. We take a DNA fragment. Those have binding site but not the protein yet. So we mix that with the protein X and DNase I, and so we can get this type of result as we see. So DNase I is cleave DNA randomly and enzyme concentration is controlled. So the each fragment cut only at a once. So DNase I, we repeating it for simplifying. Okay. DNase I does not cut DNA at site where protein is bounded or remain protected. Okay. DNA fragment are separate on electrophoretic sequence or electrophoretic gel in sequence wise. So we can compare lane A which have protein in their DNA and lane B where we remove the protein, and then we can compare where the sequence and what is the exact sequence where the DNA is binding by comparing each strand or each lane. Show absence of band in the specific region in lane A. where protein is present, so there is missing or there is gap. This indicate the protein binding site, its protection from the DNase I. Alignment with sequencing ladder allows the precise identification, and that alignment can precisely identify or precisely give us the idea about what is the exact sequence of that particular binding region. Okay, that is all about the nuclease footprinting or DNase I footprinting. In next class, we will see modification protection footprinting. It is protection type of the footprinting, but it is now modification. How we are modifying, we'll see in next class.